Hi, I'm Stacie Calad-Thomson. I'm the Chief Strategy Officer and Head of Drug Discovery at BioSymetrics. And today I'd like to tell you how we're bringing the clinic to the bench and back. The pharma industry is still plagued by high failures and attrition. Almost 90% of programs that enter clinical trials still fail, and most of those failures are due to a lack of efficacy. Having worked in this industry for almost 15 years, I've seen how clinical data and results are siloed from discovery data and how learnings aren't often shared between or even within those organizations. While there's so much opportunity to learn from real-world patient data, that data is so complex and noisy and therefore a difficult starting place to initiate drug discovery programs. Instead, we typically distill that complexity down into single protein target-based approaches that are sequential and linear. But if that target hypothesis was wrong and not widely representative of diverse patient biology, we risk spending a lot of time and money on that process before discovering a lack of translation in the clinic. At BioSymetrics, our mission is to bring the clinic to the bench and back to patients again. We initiate drug discovery programs with extensive analyses of clinical and genomic data that gives us a deeper understanding of disease phenotypes, so better precision medicines can be designed for those patients. Our approach is driven by our proprietary Elion platform, which has three components that underpin our competitive advantage: data, machine learning, and phenotypic experimental validation. We have extensive clinical and omic and experimental data from partners, public and proprietary sources. We use machine learning to then integrate that data to query and navigate linkages of disease biology and then to derive actionable insights. We then design experimental phenotypic assays in order to validate our findings, and many programs to date have been validated in vivo in zebrafish models. This approach enables us to initiate drug discovery programs with an earlier and more comprehensive understanding of patient disease biology and helps to advance the design of precision medicines with a higher probability of success for patients. Our process is not linear, but rather we can integrate new data at any stage to further enhance our understandings and progress pipeline programs forward. Our data is one of our biggest differentiators. We have partnerships with some of the nation's largest hospital networks and access to over 13 million longitudinal patient records and counting. Through our partnerships, we've systematically and repeatedly worked with partners to ingest, clean, and curate these massive data sets. And we're combining it with other private, public, and experimentally generated data to create a massive linkage of biomedical data from which to initiate drug discovery programs. The way that we're able to use that vast data is with Contingent AI, our patent-pending software that enables us to denoise and integrate diverse data. Contingent AI is an automated framework that expands the model building process, resulting in less time spent in data processing, easier data integration, and more accurate results. This iterative pre-processing approach reduces bias and helps us achieve better, faster models from the data. I've worked in artificial intelligence applications of drug discovery for the last 5 years, and I know that AI predictions are only as good as experimental validation. So the third component of our Elion platform is our phenotypic models of disease. Zebrafish have been a major focus of our platform for both data generation and prediction validations. Zebrafish are transparent, they're vertebrates, and they share 70% of their genes with humans. They reproduce in 24 hours, offering a high throughput screening model system with the benefit of whole organism in vivo biology. Based on genes that we prioritize in our data analyses, we use CRISPR to modify zebrafish and then validate the disease phenotypes. In the example on the lower right, we screened molecules in zebrafish embryos and discovered a compound that delayed development, but that delay was reversible when the compound was washed out. We've done further investigations into the mechanism of action here and are currently pursuing this program for an oncology indication. Partners. Our partnerships have enabled us to validate our Elion platform at every stage of drug discovery. We have ongoing relationships with Mount Sinai through Semaphore and with Northwell Health, where we've successfully analyzed patient records to provide clinical insights. Several of those projects have moved forward to prioritize genes and progress through a phenotype to a hit. With Merck and Pfizer, we've undertaken data-driven projects to identify the mechanism of action from a phenotypic program. In the last year, we've worked with Janssen to help identify biomarkers for clinical trial patient selections. At every stage, from clinical to gene targets, phenotypes to hits, and understanding of mechanism of action, we've enabled insights that drive projects forward. As I mentioned earlier, we have partners who have shared millions of patient electronic health records with us, clinical and laboratory test results, all in exchange for us providing actionable insights from that data. In the first example on the right, we partnered with J&J last year to predict disease severity of COVID-19 based on patient records at the time of their initial PCR test. Based on our data analyses, we've been able to identify an ensemble of biomarkers for high-risk populations and predict patients that would develop severe COVID-19. These insights were useful in patient stratification and selection for the J&J vaccine clinical trials. In the bottom right example, we've worked with Northwell Health to predict diagnoses of acute respiratory distress syndrome, both non-COVID and COVID related, among different patient populations. And we published that paper in medRxiv recently. These types of clinical insights are examples of where we can initiate more directed drug discovery programs with a deeper understanding of the underlying disease biology. Our atherosclerosis project with Semaphore is an example of this progression from data to genes to phenotype to hit. Atherosclerosis is a cardiovascular disease that currently affects millions of patients. So we trained hundreds of machine learning models to predict highly specific phenotypes and then prioritized a list of 170 genes implicated through human genomic analyses as being relevant in the disease. We did further prioritization to a top 20 list of genes and then used CRISPR to knock those out in zebrafish models. From those experiments, which included positive and negative controls, we identified a novel gene that caused significant malformation of vasculature upon disruption. We then selected compounds for a small molecule screen through an in silico derived prediction based on a library of almost 1 million compounds. And those experiments are currently underway to identify hits. As shown in the top, that type of small molecule screening prediction starts from a training set of phenotypes and associated small molecules combined with public data that's been mined. Our contingent AI associates chemical features with phenotypes and ranks compounds that are most likely to modulate that phenotype. So in a typical partnership with BioSymetrics, we may start with some clinical GWAS or omics data sets, starting from our vast database or integrating new data from our partners. From there, we will computationally prioritize gene targets based on predicted phenotypes. We can then develop and validate those phenotypes in zebrafish. Next, we use a computationally driven screening strategy to arrive at hits. Using Elion, our approach de-risks projects by starting with the patient data to derive insights that drive development of a relevant experimental phenotype, where an informed screen can be used to understand how molecules modify that disease phenotype. Following this, we have further mechanism of action capabilities to do target or pathway deconvolution and rapidly progress drug discovery programs with a higher probability of success. I'd like to finish by highlighting our leadership team that comes from diverse backgrounds with a common goal of focused on execution, delivery, growth, and solid science and technology that underpins and drives the BioSymetrics approach. We are a seed stage company being led by successful entrepreneurs in machine learning in the ad tech space. We have deep scientific and technical expertise based on academic careers. And we have industry veterans that have joined BioSymetrics from other pharma and AI drug discovery companies. We have an accomplished scientific advisory board and a growing team of talent. While we are a remote first company, we have major concentrations of employees in Toronto and Boston. Thank you for your time today. I look forward to further conversations and discussions with how BioSymetrics can partner with you to bring the clinic to the bench and back to patients again. Thank you.